Now we're going to talk about a topic that spans multiple organ systems, and it's focused on the balance of energy in the body. So we've talked about the law of mass balance as applied to other systems, for example, the regulation of the volume of water in the body. You can also apply this idea, it's simple arithmetic after all, to energy in the body, where the total energy in your body is equal to how much you have in it right now, plus whatever you take on, so through the digestive system, minus what you metabolize. And we have multiple sources of energy that come from the diet, and we can burn fatty acids and glucose and some other metabolites as well. In all cases, the energy available in molecules to produce ATP for intracellular functions needs to be balanced at a particular concentration as it circulates through the body. For example, glucose. Glucose, if in too high a concentration in the body, will produce problems. And too low a concentration, and we feel depleted and suffer from fatigue. So hypoglycemia offers insufficient energy, whereas hyperglycemia damages organs such as the kidneys, your eyes, your nervous system. So we really want a Goldilocks ideal concentration for glucose in the body, not to mention the fact that glucose is not a molecule that is as energy dense as other forms of storage. So just in terms of efficiency of storage, amount of energy per unit mass, then fats are a much more efficient way of storing energy. So our body is constantly controlling the concentration of chemicals in the body that provide a source of energy. So where does that control come from? How is energy controlled? Well, let's start by thinking about the metabolism side of this equation. So we're basically going to step through the terms of the law of mass balance and then look at the control system that monitors and adjusts the concentration of, in particular, we're going to focus on glucose. All right. Cellular metabolism. We're going to take a relatively simplistic approach to this. All the boxes and arrows that I'm going to draw out, I should say all the arrows that I'm going to draw are potentially a much more complex, or they definitely are, a much more complex series of biochemical reactions mediated by enzymes. But for our purposes, we can keep them simple. So glucose enters into cells quite readily and is then converted into glucose 6-phosphate. This is in the cytosol of a cell. Now, when glucose 6-phosphate is combined with two ATP molecules, then glycolysis can be performed. Okay, so this is a series of biochemical pathways that yields two ATP molecules and two pyruvate molecules. And that pyruvate can be used as a source of energy under a couple different conditions. But first of all, glycolysis, the bottom line here is that we made an investment of two ATPs and we got 4 out of it, in addition to the glucose 6-phosphate, which came from one glucose molecule. So with one glucose molecule, we get a net 2 ATP. Seems pretty good. Pyruvate, if oxygen is unavailable in the cell, pyruvate can be recycled by the liver to ultimately produce glucose. We're not going to talk about that right now, but just know that if the partial pressure for oxygen is low, pyruvate is converted into lactic acid, and lactic acid is then returned to the circulatory system where it can be recycled by the liver. If sufficient oxygen is available within the cell, then pyruvate is converted into acetyl-CoA. Now, this is where fatty acids come in. Fatty acids cannot be used as a substrate for glycolysis, but they can be converted into acetyl-CoA. So that is useful if there is sufficient oxygen within the cell. Because mitochondria, which are a cell's effectively power plants, can take acetyl-CoA and produce a really high yield of ATP. So that's first achieved through the citric acid cycle, which as far as we're concerned is a big circle. And that big circle will yield 2 ATP molecules. All right, so already we've taken acetyl-CoA and we've gotten as much payout from that one molecule as we got out of glycolysis from a single glucose. It also produces carbon dioxide. Finally, the citric acid cycle produces an electron and a proton, which then provides the basis for the electron transport system, which also requires oxygen. Now here's the big payoff. The electron transport system yields 26 ATP molecules. So if glucose provided the substrate, then we're left with a really high payoff. And by the way, this series of biochemical reactions explains why we breathe. Okay, oxygen is required here, and carbon dioxide is a byproduct of the citric acid cycle. That's the reason for the respiratory system and why our circulatory system transports those dissolved gases. So, the ultimate payout for aerobic metabolism for one glucose molecule is between 30 and 32 ATPs. Really good payoff. For one fatty acid molecule, it's 28 to 30 ATP. So we can see, by the way, glycolysis is often referred to as anaerobic metabolism. It has a much smaller yield, obviously, than the aerobic metabolism. And so it's quite desirable to circulate sufficient oxygen so that the mitochondria can achieve this aerobic metabolism. Okay, so you can think of metabolism as being the energetic sink. This is what cells do to generate enough ATP for all those intracellular activities that we've talked about earlier in the class. And now we're going to sort of map out where energy flows in the body. So in the cells, that's the sink. Where do we find the energy for this? Well, initially it begins with our diet. We eat fats, carbohydrates, and proteins. Fats are the most energy rich. And then we break down those molecules and either circulate them through the bloodstream or store them away. So what's in the bloodstream we can think of as being a pool of energy, like glucose. Glucose will circulate through the bloodstream, and then you'll find glucose distributed throughout the body within the cells. Free fatty acids, as we've just seen, also provide a source of energy. So cells primarily metabolize glucose and fatty acids for energy. Proteins are broken down, and they can ultimately be broken down into the form of amino acids. And amino acids can also be converted into glucose. We'll get into where that occurs in a moment. So we have the digestive system providing these pools of energy. And we've already touched on the fact that glucose at too high a concentration can be damaging to your organs like your kidney, your eyes. And so we don't want to just break down carbohydrates and have them circulating around in the bloodstream. Same thing with the free fatty acids. It's among other things, not a very efficient way of storing energy. So glucose can be fairly readily converted into a larger molecule known as glycogen that occurs within the liver and in skeletal muscles. Free fatty acids are packaged into fat, stored in fat cells. And then glucose can also be stored away into fat in those adipose cells. So excess glucose is first stored as glycogen. We have limited capacity for glycogen storage in our body. It's available in the skeletal muscles. You can increase your capacity for glycogen storage through exercise. Since that's an intracellular form of storage, energetic storage, then it's convenient within the skeletal muscles to be able to convert glycogen into glucose when you're exercising. Amino acids, of course, can be built up through the synthesis of proteins. And you can think of proteins, even though they provide a structural role, as ultimately being a source of energy if the body needs it. So when the body needs energy, then glycogen can be broken down, proteins can be broken down, and so can fats into fatty acids. Okay, now we're going to turn our attention to the control of glucose. So these pathways, by the way, all these arrows do correspond to pathways that can occur in particular cells in the body, and those cells reside within muscles, fat cells, the adipose cells, and in the liver. Now, these pathways don't occur in all of those organs, but the conversion of glucose into fat does occur in all of them. The ability to convert amino acids into glucose occurs only in the liver. Glucose is stored as glycogen in the liver and in the skeletal muscle. So these pathways represent efferent actions for a control system. Okay, the goal here is to deliver a certain concentration of glucose in the bloodstream. And you can think of the pathways that draw from the glucose pool as ways of storing. These are pathways for reducing the concentration of glucose through storage, and then glycogen, the conversion of amino acids, and for that matter, the generation of fatty acids too, are ways of essentially cashing in that saved storage of energy. Now, where is the decision making about the homeostasis for glucose made? Thus far, we've considered a variety of control systems in the body. They seem mostly to reside in the brainstem. This is an example of a control system that doesn't. In fact, the central nervous system doesn't play any role in this control system that we're going to map out. Where is it? Well, it's vaguely associated with the digestive system. The pancreas has cells that serve this function. And we've already talked about how the pancreas plays a role in secreting digestive enzymes, as well as a bicarbonate to neutralize the acidic chyme that emerges from the stomach. So it has those digestive functions. This function is kind of separate. It's obviously related to digestion because of the way that food provides a source of energy, but it doesn't actually produce any products that make their way into the digestive tract. Okay, so let me be more specific. We have two types of cells in our pancreas. They're called alpha and beta cells. And they both serve as sensors and integrating centers for energy balance with a particular focus on glucose. The beta cells secrete insulin, which stimulate energy storage. Okay, that's what insulin does. Another hormone which you might not be familiar with is called glucagon, which is secreted by the alpha cells, and glucagon does very much something the opposite of glucose. So instead of storing energy, it stimulates the release of energy, or I should say, the conversion of molecules into glucose so that it's available in the bloodstream. So let's consider some specific examples of this. So let's just monitor plasma glucose. And here we see that after you've eaten breakfast, you get this increase in plasma glucose. And the magnitude of this increase depends on the nature of the meal. If you eat a bunch of sugar, then you're basically going to have a huge spike in glucose. If you eat, eggs and toast or something like that, depending on the nature of the toast, you might have a more gradual rise if more complex carbohydrates take longer to absorb. Now, your beta cells in your pancreas respond to this increase with a surge in insulin. That's the signal to the body that this glucose should be stored away and not circulated at high concentrations in the blood where they can cause damage. And by storing away glucose, that allows your body to potentially cash it in later on. Okay, this is a way of storing energy so that you don't burn it all up right after breakfast. Now, this pattern gets repeated in the subsequent meal. So for lunch, you get an increase. It might be a larger meal. And then notice here that after each meal, there's initial increase and then a leveling off. Okay, so that leveling off is indicative of homeostasis, okay, a negative feedback loop. And that is largely being driven by the insulin. Now, the exact shape of these curves depends on the nature of the food and how well your beta cells are functioning. Then at dinner, you might have a very large meal, and then you begin your fast that you maintain throughout the night. Glucagon has a less predictable pattern of changes in concentration. Glucagon essentially increases with fasting. So you can see it the best here around dinner time. Before the meal has been eaten, you can see that increase in glucagon. But glucagon doesn't have as rapid a temporal response to these events, and your body actually pays most attention to the relative concentrations of glucagon and insulin. So insulin has the most rapid influence on the concentration of glucose in the body, and then glucagon has a more sort of long-term gradual influence. And again, it's the relative concentration of glucagon to insulin that matters the most. All right, so that's the output of our control system. And we already know that we're talking about alpha and beta cells, so let's map out that control system. Now, those biochemical pathways that we see up in the upper right can be included on the efferent end of our control system. Now, insulin release, as I've mentioned, causes your body to store away glucose in the form of other molecules that are better for storage. So in the liver, we have glycolysis. So some of the glucose is converted into ATP through that mechanism. The liver has some limited capacity to store glucose as glycogen, and then also it produces fat. The fat cells of the body, the adipose tissue, also undergoes glycolysis, and in response to insulin, there's a conversion of fatty acids to fat. Finally, muscle has a variety of means of sequestering glucose. So first of all, it burns some to produce ATP through glycolysis. We have glycogen stores within the skeletal muscles. Glucose is stored away as fat, and you produce proteins in response to insulin. So that's a conversion of amino acids into proteins. Now, all of these efferent actions serve to counter plasma glucose. Okay, they act to reduce plasma glucose concentrations. And so these are activated in a fed state after you've eaten a meal. Glucagon, on the other hand, acts primarily on the liver. And it's there that glycogen is converted into glucose, made available into the bloodstream, and amino acids are also converted into glucose as well. So both of those contribute to the plasma glucose concentration. Now, in order to sense and respond, or to make this a control system, then we have to add the afferent and integrative portions of our control. All right, so we have the alpha cells, which are responsible for the glucagon release, and the beta cells, which release insulin. And this flowchart maps out essentially the answer to the question that we set out to address, which is how is energy controlled? But before we conclude, there's a disease that I want to talk about, which is diabetes. And we don't often talk about many diseases, but in this case, it's quite useful to think about how diabetes squares with this control system that we've mapped out. So first of all, there's type 1 diabetes. And type 1 diabetes is an autoimmune deficiency, and this accounts for just 10% of diabetes cases. And because the consequence of this autoimmune deficiency is that it kills the beta cells in the pancreas, then we essentially lose the ability to reduce plasma glucose concentrations. So this results in hyperglycemia, which is quite dangerous. And so we can compensate for that by insulin injections, essentially replacing the function of the beta cells by monitoring plasma glucose levels and having regular injections after meals. And that essentially rescues the functions, even though it doesn't work as well as the natural presence of the beta cells. It effectively recovers all of these pathways so that your body regains the ability to reduce plasma glucose concentrations. Type 2 diabetes, which is 90% of cases, makes cells less sensitive to insulin. This is a little bit more insidious in terms of treatments, although it can be less severe in its effects on the body. It's created by making the cells less sensitive to insulin. So these pathways are reduced. And this is linked to lifestyle. 80% of type 2 diabetes cases are in people who are obese. And it's also linked through obesity or indirectly to diet and a lack of exercise, a sedentary lifestyle. So type 2 diabetes is a real epidemic for Americans given our relatively sedentary lifestyle and our poor diet. Now, the treatments are quite varied. There's a variety of drugs, but lifestyle changes are generally recommended as a treatment for type 2 diabetes.